We've got a couple of plant breeding sessions on the program. The first session, plant breeding for organic farmers, number one, Back to the Future. And we have three speakers. Our first speaker is Walter Goldstein from the Mandamin Institute. And Walter's been breeding corn for 25, almost 30 years. Come on up.
The Mandamin Institute is located in southeastern Wisconsin. We're a nonprofit organization. We do different things, but our particular focus is on breeding corn for organic farmers. We have a bit of a different vision for corn. One thing, of course, is we're concerned about the systems in which corn are produced, that they're healthy systems, and we want to encourage healthy soils, healthy management practices, but also we're concerned about the breeding of corn and where the seed comes from, and that it actually fits the needs of organic farming. We do have a nascent program. We call it the Open Pollinated Breeding Network, that we're working with farmers to improve field corn. And we also have somewhat of a different attitude about corn, rather that corn is a partner in the breeding process rather than being a slave or a machine. And in terms of the way that we want to take corn, we would like to enhance the natural gifts that it has, in particular of nutritional value, and try to couple that with yield, so that we get something rather than just a downward spiral of yield at the cost of quality, that we try to combine quality and yield. For organic farmers, what's really important is, as we've heard in the previous session, is not getting contaminated with transgenics. And it is possible to breed corn so that it won't accept pollen from transgenic corn. We also are working with breeding nitrogen efficiency into the corn, in particular working with corn that works together with bacteria to provide nitrogen to the corn. And also I'll speak about some trials that we've done with competitiveness with weeds, which are an issue for organic farmers. Ultimately, our output are inbreds, hybrids, but also open pollinateds for farmers. We're part of an OREI team. It's funded by nifH, USDA. It includes Paul Scott, who's my compatriot here, and also Margaret Smith from Cornell, Jody Edwards from USDA, also in Ames, Iowa, Kevin Montgomery Consulting, and Rich Pratt from New Mexico State University. And I run a standard inbred selection program. However, we do utilize organic criteria in our selection. So we're selecting for traits that really are important for organic farmers. We do have a summer nursery in Wisconsin, and to my knowledge, we have the first organic winter nursery in Puerto Rico. We've run it now for 4 years. Actually, this would have been our 5th year, and we routinely can produce seed crops. So we have 2 growing seasons in a year in order to advance our breeding. We do breed with our own cultivars. We do make improved synthetic populations by combining our better inbreds or breeding lines. We do use PVP, ex-PVP inbreds, and we do use plant introduction landraces a lot in our breeding in order to get some of the traits that we need for organic farming. This is an example of some of the selection that we do. We select under nitrogen. Our nurseries are generally under nitrogen limiting conditions. We go through our nurseries, thousands of plots, and we actually count emerging plants, measure their height, and give them a color score. And on the basis of those numbers, we decide who gets pollinated. And that's an important screening for us in order to be able to develop the kind of vigorous plants that are going to compete with weeds and give reliable emergence and get nitrogen, however they get it. Yields, I'm not going to go greatly in depth into it. We do annual testing in Wisconsin. We've generally had 2 sites with USDA in Iowa in order to be able to screen hundreds or thousands of potential hybrids. We also do head-to-head testing with the U.S. testing network on states across the eastern portion of the country. We usually test in 3 different maturity zones, and we're comparing head-to-head with other breeders and with a lot of conventional hybrids. And some of the hybrids that are hours by hours, where we're crossing our own inbreds with each other, have looked competitive. However, we really need to have more data in order to make a release. So we're still waiting for that 3rd year of data on some of these things in order to know which direction to take things in. We also have found that our inbreds crossed with commercial inbreds do yield very well, some of them, and the problem is that the quality that we're breeding for diminishes when we cross our inbreds with other inbreds. The hybrid grain quality goes down. Carotenoid selection has been an issue for us, and we haven't done too much more than visual selection of seed and focusing on breeding first off with populations for carotenoids, and here you see some HPCL data with retinoid active carotenoids, and how we have improved it from a commercial hybrid, which was a well-colored hybrid. Some of our populations here have considerably higher carotenoid contents, even double. And then, of course, we try to focus that into inbred development with a high carotene content in the corn. The target for that, of course, is a human being, but through the egg. And we have these fabulous eggs, just fabulous. I've turned into a total snob about eggs. And the chickens do seem to, my impression is they do seem to prefer orange kernel corn. And when we slaughtered some of our layers on my farm this year, we saw they seem to be almost painted orange on the inside, accumulating those carotenoids in their fat and putting those into our yolks. We did a couple years of weed competition studies with hybrids and open pollinated corn coming out of our program, and compared them with some commercial hybrids. And first we did a study in 2002 with giant ragweed, and the giant ragweed was planted, 1 plant of giant ragweed, 1 plant of corn, 1 plant of giant ragweed, 1 plant of corn. And we found that if we made hybrids with a population called Nokomis Gold, which was one of the populations out of our breeding program, that it reduced weed yields by about 1/3. And that was, and Nokomis Gold itself was very capable of reducing the yields of the ragweed. We also, later on in 2006, we did a study, a much larger study with hundreds of different hybrid combinations and open pollinateds and top crosses between open pollinated corn and inbreds, so-called top crosses. And we looked at weeds. We actually took a patch of each plot and we planted sunflowers into the corn. And then we weighed the sunflowers, and we also did weediness scores in the plots. And we did find actually large differences between the conventional hybrids. They yielded the best, but they had a much greater weed scores and greater growth of the sunflowers in the stands. So there's something going on there. We have not really discovered how the corn does it. I suspect it's not necessarily just foliage and shading, but that there's signaling going on in the ground, because we did do some light measurements and weren't able to correlate that with weediness. A crossing compatibility, I mentioned these are traits that we're utilizing. We're using several genes that are found naturally in corn and in land races. And we're using them to build a strong barrier. We're putting them into inbreds, and we've recently had quite a bit of interest in producing an open pollinated that would have also those traits in it, as well as nutritional value. We're working together with USDA, with Paul Scott's lab, who's developing a marker system for one of the genes, GA1S, and also with North Carolina State University with Major Goodman. I had an almost ecstatic experience this summer when I went out to my test plots, we had taken rows of inbreds and hybrids that we had developed that we had been testing for years to get the trait into the corn and very active. And we took them and we detasseled them, and we had a male that would pollinate them that did not have the same genetic system. And the genetic systems work by if the corn has the gene, say GA1S, and the pollen that lands on it doesn't have that same gene, it will not allow the pollen tube to grow down the silk into the little germ. So, and we had actually fabulous results out of the total number that we put out here. We had 35 that had no pollinations, 6 that had just a few kernels, and 4 that had occasional ears that were pollinated. So I was gratified to see it would work, but on the other hand, that's 1 year. And you just don't know how stable that's going to be from 1 year's worth of data. Another area of interest is in nitrogen efficiency. In 2009, I had a field where I was growing a set of landraces in corn that we were interested in for quality purposes, and we had a slew of different inbreds there, both from our program and also from a commercial program that we were working with. And the site happened to be nitrogen deficient, and the conventional inbreds and our inbreds all looked deficient in nitrogen. However, and we had inoculated all the seed, which we do inoculate with a suite of nitrogen-fixing bacteria. But it didn't. These particular land races, some of them, they looked like they had been highly fertilized with nitrogen fertilizer, and the field had no history of fertilization for at least 4 years. And the differences were to the row. Well, we looked at the isotopes, the natural isotope composition in the plants, and using delta N15 ratios, it appeared that some of these races may have fixed as much as half of their nitrogen from the air. So we made hybrids with those races, and we grew them out in 2010 in an unfertilized plot. And we compared them with a set of 4 different commercial organic hybrids. And what we found was a difference in protein content, in the yield of the protein, that there was more protein being harvested with the nitrogen efficient corn. In 2013, we took we were making selections from those hybrid crosses and screening them. And by 2013, we put them on a field which had been in, it was in a low soil organic matter soil, 1% sandy soil with clay underneath, and well stocked in potassium and phosphorus. And we found quite a bit of deficiency in nitrogen on that field. All seed was inoculated. We had some checks in there, and we replicated our breeding lines twice. And what we discovered when we looked at the checks, and those were checked populations and one breeding line that did well under normal conditions, but they did very poorly. We had particularly barrenness. We had only 19% seed of the plants set ears on the checks, and of our top 30 lines, we had 75% of the plants setting ears. Well, that was actually pretty shocking and remarkable and interesting, but not conclusive, of course. Jerome, I'll take questions later, okay? Unless it's one of clarity? Is it one just didn't understand? Okay. This is so we continue inbreeding and these lines and taking a look at them. And this year, because I don't have much seed to look at these things, I started putting in checks. And here you can see checks. We had 2 pieces of land. One had been in spring wheat and was well supplied with nitrogen in the soil profile. And down below here we had corn following after corn, no fertilization. And these are the same PVP lines. These are old commercial inbreds, Lancaster and Iodent and a B73 type for those who are in the know. And we could see actually rather striking differences in terms of how these plants were thriving under such conditions. Whereas right next on the corn after corn ground, we had some of our inbreds that actually looked very, very good and didn't look nitrogen deficient. Happens to be a cross between PHK 76, which you saw on the previous slide, with a Mexican landrace and another B73 type cross with a landrace. So we're seeing these differences. So we're beginning to wonder, how is this thing working? And I sent my colleague Lucas Nabert at the Institute of Ecology at the University of Oregon does more work with genetic markers, and we sent him some seed of some of our different lines. The question is, in part with this seed, if the plant is having a different relationship with endophytic bacteria that can fix nitrogen. And endophytic bacteria now are kind of a hot subject in plant physiology, as they're implicated in producing much of the hormones that plants utilize, and in regulating growth and having all sorts of effects of protection and nitrogen fixation and hormonal regulation. So endophytes are a hot subject, and Lucas surface sterilized the seed and extracted DNA from the ground up seed to look for nitrogen fixing genes and fungal specific genes. In particular, we were interested in Fusarium, as we have speculated that Fusarium might be a particular Fusarium verticillioides might be a gatekeeper, actually a very dominant endophyte in the plant, which prevents some of the beneficial bacteria from being able to operate in the plant. And here's some preliminary results with the diazotroph, the nitrogen-fixing bacteria, the nifH gene, the protein which fixes nitrogen in the plant. And you can see here that we have, and I don't have a pointer, so I'm going to step in here, the PHK 76, 1 year under organic production, PHK 76, multi-years of organic production, PHK 76 crossed with the landrace. You saw the corn in the last slide. You can see here there was a significant increase in the nitrogen, the nifH protein genes in that particular treatment. And here we can see with crosses between PDF, which was one of our inbreds, a stiff stock type, and crosses with the Peruvian, and crosses again with the Mexican, where we saw a tendency towards having more of the nifH gene in the seed. Looking at Fusarium verticillioides, we see that if we look at PHK 76, 1 year, multi-year, and the PHK 76 by the Mexican, we really don't see much difference in the verticillioides. However, if we look at the PDF and then crossing it with some of the putative nitrogen-efficient corn from Peru, we do see a drop in the verticillioides that's present there for some of those combinations. Now, if we look at total amount of fungus in the seed, fungal endophytes in the seed, and here we have PHK 42, which had been conventionally grown, and PHK 42, which was organic, and PHK 76 with 1 year organic, and PHK 76 with multiple years of organic. We did have a significant tendency for the corn, which had been more under organic production, to have more total fungal biomass or fungal DNA in the seed than we did with the conventional, but with no difference in the verticillioides. So it wasn't because there was more of the toxic fungus that was accumulating. So we're seeing some shifts going on. Whether these will stand more research or not, I'm not sure. Here we can see AR-guard population here segregating for nitrogen efficiency. Does that mean it's segregating for partnerships? And that's a question that I think is important. Overall, I can say that I think through our breeding program, we keep on seeing evidence that adaptation happens, and it happens whether we're doing it consciously, whether we're doing it through selection, or whether things are happening in the plant, the plants are rearranging under the conditions which we're providing them. One of the things that we have noticed is that culture shock. We talk about bringing in inbreds from the outside, from conventional nurseries. We often see under organic conditions that they don't do well for the first year. But after we keep them and grow them for several years under organic production, they seem to thrive more, in particular better nitrogen utilization. And we're not quite sure what's going on there, but we also do see when we take some of these inbreds and work with them that we have a certain level of phenotypic plasticity, whether it's an inbred or it's an advanced breeding line from another program. We select for the more vigorous ones, and we have seen gains in earliness and also in chlorophyll content indicating greater nitrogen efficiency. And some hybrids that we've made by crossing these more apparently better adapted selections seem to produce similar yields as to if we would have taken the original parents and crossed them together. So here you can see an example of that. This is M632, which is actually a BC5. We got out of an A632 type, and it's taller and darker green than the original cultivar which we received from Minnesota. Protein quality, our initial, let's see, how much time have I got? 2 minutes? Okay, that went fast. Did I really? Okay.
I'll give you 3.
Okay. We did initial feeding trials with Flowery 2 corn, and we discovered, which was reputed to be high in methionine, as we're breeding corn for high methionine content. And we did find that it would replace, we worked with Organic Valley in the University of Minnesota. We found it did replace synthetic methionine in terms of there were no difference or little difference. There was a 2 to 5% reduction in egg production when we compared it with a normal diet with synthetic methionine. This is an issue for organic producers, is the use of synthetic methionine. It has been linked with IGF-1 production in birds. IGF-1 is a growth hormone, which isn't good for us to be having in large quantities when we're middle-aged people. It's been linked with cancer and type 2 diabetes. So there is a reason for us doing that. However, the Flowery 2 gene did condition a lower seed size and lower yields, and we couldn't get around that. We tried it in different backgrounds. So we went away from it and we started selecting opaques out of our own breeding program. I'm not going to have time to get into that right now, but these opaques have been a bit of a mystery to us. They seem to appear. We suspect that there's epigenetic regulation of the trait of opacity based both on our own experience and the literature. And I think I'm rapidly running out of time, so I'm going to have to go to skip that. But
Walter, you have 5 minutes if you don't want to take any questions.
Okay, very good.
But I know you want to take questions. So.
Okay, so let's I can go back to that as questions.
Go.
Then those the mysterious appearance of opaques in our breeding program, and that they are associated with a higher methionine content, a reduction of the alpha zein, which is a major protein in corn, which makes the kernel hard, but also is low in protein quality. Well, we found these opaques that produce a softer kernel and have lower production of the alpha zein, but higher production of higher quality nutritional proteins. And these, as I said, these opaques have appeared in our breeding program. Here you can see PHK 42, which is a standard pioneer inbred. And you can see if we outcross it with something, the purple plumule, which is uniformly found in iodent inbred lines, disappears. Here you can see one of the selections out of PHK 42 for a little bit more vigor. And out of this 11.2, we selected 11.2.1, 11.4. 11.2.1 was opaque, maintained the same purple plumule. If there had been an outcrossing event, it should have turned color. We found it with our advanced gameta cross-incompatible lines, also these opaques have been popping out in them as well, and they have the ability to control pollination. So we suspect that there's a level of epigenetic control on that trait, and that when we're working with breeding corn, that for organic farmers, that there's a kind of an emergence of traits that can occur. It may be stimulated by the kind of conditions that we're exposing the plants to. We suspect either shifts in the microbial populations in the seed and in the plants associated with organic production, and that's also part of the equation. It may that we may be working not only on a genetic level, specifically with DNA, but we may be working on the regulatory level and the plant's ability to shift around its program and its body. So that's all very exciting, but as the proof is on the plate, and the point is really with this corn is to get it on, not just on my plate, but also on your plates.
We do actually have time for a few questions if you have one.
Let me get it.
So how is your corn available? How does it wind up on my plate?
How does it wind up on your plate? Well, if you want the corn now, the way that you can get it is in the form of an open pollinated. We released one hybrid this year in cooperation with a local farmer. That seed's all gone, and we have not produced another hybrid. But we do have plenty of open pollinated corn. If you were to want to get the corn, you would have to sign a contract. The contract would stipulate that you could grow that corn on your farm for the rest of your life without having to pay us anything. But if you want to sell seed of that corn, then you have to go back to us. We have to know about it. The person you sell the seed to has to have a contract with us, and we want a royalty.
So do you have plans to release this corn through seed companies?
Absolutely.
And timeline on that?
That's something. So basically, we have three organizations involved in a kind of a triangle. We have the Mondawmin Institute, which does public breeding. We have Nokomis Gold Seed Company, which intends to take the hybrids through very rigorous testing and to develop them. And we have a third company run by an entrepreneur who will invest in the breeding process in exchange for exclusive rights for a certain period of time for hybrids. That's a different model than for the open pollinated. The open pollinated is going from farmer to farmer. The hybrid is going are going to be going through, probably, we're hoping, if we can pull this off, through a series of events that will attract investment. Investors don't want to invest in something unless they have some sort of guaranteed return.
Return.
Yeah, return.